One very exciting finding was announced for patients with metastatic hormone receptor positive HER2 negative breast cancer with mutations to a gene called PIK3CA. These mutations can cause breast cancer to be very aggressive and difficult to treat. To find better targeted treatments for these patients, the phase 3 INAVO 120 trial tested a new drug called inavolisib, which targets PI3 kinase in combination with palbociclib and fulvestrant versus palbociclib and fulvestrant alone in the control arm. This trial was specifically tailored to patients with metastatic hormone receptor positive breast cancer that have a mutation in the PIK3CA gene. Researchers found that adding inavolisib to this treatment regimen led to an incredible 15-month progression-free survival compared to just 7.7 months with palbociclib and fulvestrant alone. This is a 7.3-month increase in progression-free survival, which is absolutely huge for patients. This is a big win, and we're really excitedly following these findings. At SABCS this year, we also heard the first results from the phase 3 HER2 CLIMB-02 trial, which tested a new combination of HER2 targeting therapies, tucatinib and TDM1, in the second-line setting of metastatic HER2 positive breast cancer. One important thing to note about this trial is that about 43% of people in this trial had brain metastases. And this is really unique because most clinical trials will exclude people who have brain metastases from being included. Living with brain metastasis is a really difficult thing. It can decrease someone's quality of life, and it's really difficult to treat people with brain metastases because of the blood-brain barrier that prevents most treatments from accessing these brain metastases. But results from the HER2 CLIMB-02 study showed that patients had a median of 7.8 months before their cancer progressed, compared to 5.7 months alone for those who were only taking TDM1. This means that even patients who had brain metastases were able to receive some benefit from this treatment. In light of these results, we may have new options available for people who are living with metastatic HER2 positive breast cancer. If you want to learn more about HER2 positive breast cancer, check out our last episode of Breast Cancer Breakthroughs to learn more about the treatments that are available and what's left to come for research discoveries. Right now, immunotherapy is only an option for patients with triple-negative breast cancer. But some of the findings from this year's San Antonio Breast Cancer Symposium may change that. Two phase 3 clinical trials, the CheckMate 7FL and Keynote 756 clinical trials, are testing immunotherapy drugs in people with ER-positive breast cancer. These results were also preliminarily announced at the ESMO 2023 Breast Cancer Conference, which you can read about on the Komen blog, linked below. But updated results were presented at SABCS, and this shed some light onto which patients were really most likely to derive the most benefit from immunotherapy in the estrogen receptor positive setting. Researchers found that tumors which had high levels of the PD-L1 protein and low levels of estrogen receptor were the most likely to receive benefit from this treatment. We spoke with Komen researcher, Dr. Justin Balco, who is looking forward to what's in store for immunotherapy for estrogen receptor positive breast cancer. And he discussed how he hopes clinical trials will be designed to get these drugs to the right patients.
There was a number of different studies that were updated from ESMO this year on high-risk ER-positive patients and the impact of immunotherapy. So immunotherapy is something that my lab works on, trying to identify the right biomarkers for patients. We've developed some really clinically useful ones that we're going to, I think, get the opportunity to deploy in ongoing clinical trials in this setting. So I think that, I have always been very interested in the triple negative breast cancer space because that was the most obvious place that there was the best chance of likelihood that, or the best likelihood that immunotherapy would be effective in triple negative. But as we know, as aggressive a disease it is that big of an impact that can have on the breast cancer field as a whole. The majority of our patients are ER positive. And so seeing it work, even if it's in a subset of patients, which we don't realize yet, is really, really encouraging. And so I think that there's a huge opportunity for impact. What I do hope, and that I see in the field that could be a problem, is right now immunotherapy is approved in all triple negative breast cancer patients. But we see really only a benefit of 10 to 15%, maybe even in terms of event-free survival, which means we're massively overtreating triple negative breast cancer patients with these drugs that can cause permanent and or life-threatening toxicities. And so once we've done that, it's really, really difficult to go back. So this reinforces that we need biomarkers going forward. And that can be a really tricky thing to do when those phase 3 trials that lead to approval are being guided, managed, and sort of guarded by industry. And so I think as a field, we need to be really careful, particularly given the fairly small benefit that we see in triple negative breast cancer and now seeing in ER positive breast cancer, but very clinically meaningful, of making sure that we're not just doing exploratory biomarker analysis for manuscripts and for scientific reporting, but we're really taking the biomarkers that have shown activity in other tumor types, in breast cancers, in the literature, and making clinical assays out of them and being proactive about the way that we test them and integrate them into our ongoing clinical trials, or else we could be in a problem area of immunotherapy gets approved for all high-risk ER-positive breast cancer patients, and we don't have a good way of selecting patients that are most likely to benefit because we can't do those de-escalation trials.